In this module, we'll talk about cytogenetic disorders. Cytogenetic disorders basically are the disorders in which there is a structural abnormality with the chromosome. You are familiar with what chromosomes are, and these structural abnormalities are generally visible through a microscope. In order to detect cytogenetic disorders, a technique called karyotyping is used to diagnose these cytogenetic disorders. Karyotyping is a photographic representation of G-banded stained metaphase chromosomes spread in order of decreasing length. I'll show you a karyotype in the next slide. Here I would additionally like to point out that these chromosomal abnormalities, cytogenetic abnormalities, are roughly in 1 in 2,000 newborns, and 50% of the first trimester abortions have chromosomal aberrations or chromosomal abnormalities. As promised, here's a karyotype. You can see that we have 46 chromosomes, which I mentioned earlier too. Out of these 46 chromosomes, 44 are the autosomes, meaning that these chromosomes do not carry any genetic information that plays any role in sexual differentiation. The sex chromosomes are X and Y. Men have one X chromosome and one Y chromosome, whereas females have two X chromosomes. So if there's an abnormality, we'll look at some of the abnormalities chromosomes can have later. But here I would additionally like to point out the chromosomal nomenclature. First of all, we look at the position of the centromere. We know what that is. We have already learned about that. Chromosomes can be classified into three different groups for human chromosomes. They are metacentric, meaning that the centromere is in the center of the chromosome. That is metacentric chromosome. If centromere is not exactly at the center, so the two arms of the chromosome, I will point those two arms out to you here, just a second. So they are the two arms of the chromosomes, P arm, P stand meaning petite, and the Q arm, which is the next alphabet in ABC. So petite arm, two arms, if both the arms are of the same length, the chromosome is metacentric. If the P arm is smaller, relatively much smaller than the Q arm, these are submetacentric chromosomes. And the chromosomes in which the centromere is pretty much at the tip of the chromosome, they have a very, very small P arm. These are called acrocentric chromosomes. There are other two types of chromosomes too, but they are not in humans. I'll just mention there are telocentric chromosomes, which are generally in mouse, and holocentric chromosomes, in which the whole centromere is present all along the length of the chromosome. We don't have those chromosomes either. Now, P arm and Q arm are divided into arbitrary regions. You can see by arbitrary, I mean hypothetical, theoretical, that scientists have just come up with these regions. 1, 2, 3, you can see that, for example, in this chromosome 1, you can see that these regions are marked right here. These regions have bands, and these bands, if we actually allow these chromosomes to stretch, I won't get into how we do that, but if the chromosomes are longer, even subbands are visible. So if we want to refer to a particular region of the chromosome, we will first say the number of the chromosome number 1, for example. Then we will say which arm it is. So let's say we want to talk about this particular band here. So it will be chromosome number 1. And then we will say the Q arm or the P arm. In our case, it's the Q arm. And then we will say, okay, so which region this particular band is. Then we write the region number and then the band number. So this is how basically you refer to a particular region of the chromosome. Now let's move forward, and we will talk about chromosomal abnormalities, types of chromosomal abnormalities. An exact multiple of haploid, which is N, which is the number of chromosomes in a gamete, a sperm or egg, is exact multiple is called a polyploid. 2N is the number of chromosomes in our autosomal cells or normal cells, non-reproductive cells. So polyploid organisms or polyploidy is basically referring to complete addition, an extra set of chromosomes. So if someone or some organism has 3 times the N, it will be called 3N, or 4 times the N, it will be 4n. That is polyploidy. A chromosomal number that is not exact multiple of n is called aneuploid. Aneuploidy can result from non-disjunction of homologous pair of chromosomes in first meiotic division or failure of chromatids to separate in second meiotic division. This can result in monosomy or trisomy, 2n-1 or 2n+1 respectively. I would just like to remind you again, here's the meiosis, that here we see meiosis in which there is non-disjunction, and you can see that some of the gametes, two gametes have extra chromosome, these gametes have an extra chromosome, and these gametes have one less chromosome. So this is a type of aneuploidy. This would be n, this would be n-1, 2n-1, and this would be 2n+1. So now let's move forward. Let's talk about the chromosomal types of structural abnormalities of chromosome. There can be translocations, meaning part of a chromosome breaks and it exchanges a similar size piece, or the piece can be actually longer or smaller. A piece from chromosome breaks and fuses with another chromosome, and there is another piece from here. You can see on the, for example, in the photograph, a little piece of blue chromosome is being exchanged by a little piece of with a little piece of red chromosome. So this is balanced reciprocal translocation. There's another type of translocation in acrocentric chromosomes, which is displayed in the right beneath it. In this, as I've said, that acrocentric chromosomes have a very small P arm. So here you can see the P arm is very, very small. If a translocation occurs between acrocentric chromosomes, two types of chromosomes are formed. One is a large chromosome, other is small chromosome. Generally, the tips or very tip or the small arm of the acrocentric chromosome is coding for RNA for ribosomes. And since we have more than one acrocentric chromosomes, I can show you that later. We have 5 acrocentric chromosomes. If this small chromosome is lost, it won't have any effect. So anyways, this is a centric fusion or Robertsonian translocation, that's what it is called too, between two acrocentric chromosomes, giving rise to a larger chromosome and one smaller chromosome. There is next category is deletion. A little piece of chromosome is broken off and is lost. That is basically a deletion of chromosome. Another way we can have chromosomal aberration is the two tips of chromosome break and chromosome becomes a circle like shown here, and these two tips, when they break off, the chromosome becomes a circle and the tips or the fragments are lost. So these are some of the types of chromosomal aberrations that can happen. Let's look at some other. Isochromosome. Normally a chromosome, as we know, when during meiosis it separates along this axis. In some cases, a chromosome, if a chromosome separates two chromatids, rather than two chromatids separating, the two P arms go to one chromosome and the two Q arms go to another to the other chromosome. We have a chromosome with identical P arms, both arms being identical. This type of chromosome is called isochromosome. This generally happens with the X chromosome. There are inversions. Lastly, a piece of chromosome, we have a chromosome, a little piece breaks and it inverts 180 degree and then again fuses with the chromosome. This is the inversion. If it involves the centromere region, it will be pericentric inversion. If it does not involve the centromere region, it will be paracentric inversion. So we will continue our discussion of cytogenetics in the next module.